4. Okay, I believe we are live now. Hello, everyone. Good to see you guys. It's been a little while. Well, today I'm on with botanist Dr. Buzgo. Hello, how are you doing?
Hello, everybody.
And we're going to talk about stuff and things. Lots of stuff and things today. We have a long list of things. We'll try to get to all of them. Um, but first of all, tell us a little bit about your background and research and that kind of stuff.
And in that part, it became part of a longer question: where do flowering plants come from? That is known as the angiosperm phylogeny group. And I had the privilege to be a PhD student and a postdoc student right in the time when the evolutionary history of flowering plants was um, uncovered or discovered using DNA analysis. And then at the same time, there's another project called Deep Green, where people looked at the deep evolution of green algae, where from then flowering plants or land plants did evolve from. I was not involved in that directly, but one of my supervisors was in it, and I kind of was orbiting around it. With that, I was heavily exposed to things like phylogeny, and later then, as I assumed a postdoc position, I was involved in gene expression studies to actually answer the question: when we have genes on one side, how do these genes then relate to the shape or the features that we see? So how do these genes make that, for example, flower has petals, or how do these genes make that teeth grow in our mouth and not in our ears?
Mm-hmm.
Questions like we
Important
have
things, yeah.
ears, why do we have them in our mouth? And so that went in that direction. From that, of course, the evolutionary question came up. Now, does I teach biology, or on that side, the entire beginning of life is one of the big questions that I also teach. And of course, then with that, I get exposed to this and get more interested and develop more thoughts.
Yeah, with the study of why do things develop where they are, you're kind of like a plant evo devo kind of guy, huh?
Exactly. It is plant evo devo stuff. That's what I did in my postdoc, yes.
I think I think the evo devo stuff is really fascinating because it's kind of the intersection of historical evolution with practical testing. We can tweak these genes
Mhm.
and figure out what is the result and how does this factor into the evolutionary history of this lineage.
And you can see how genes interact with ecological adaptation.
Mm-hmm.
You have a flower that is like, if you look at columbine, the Aquilegia flowers. So there are some species that have extremely deep spurs that are adapted for certain butterflies or hummingbirds. And there are others which are very open and shallow, adapted for flies, for the big pollinating flies, and short-tongued bumblebees. And how does this adaptation happen? Which genes are responsible to do that? And so in my background, actually competing, a rivaling team from Harvard was working on that topic, and they're still working on it. And it was very interesting to actually see which genes can lead to that adaptation. Yes.
Yeah, there was a paper that came out. I think it was late last year on a particular gene controlling the inflorescence. Which, if you're wondering what an inflorescence is, people, it's like when you have a bunch of flowers that are kind of clustered together, and there was they were talking about this gene which, if you could change the timing of this gene, so you could do heterochronic experimentation with it, you could generate the whole diversity of inflorescences.
Yes, and you can even flip an inflorescence into one big terminal flower. And that gene is called terminal flower. So that's what it does. It's when that gene is broken, then instead of having a nice candle-shaped inflorescence, then Arabidopsis forms one odd-looking terminal flower, and that became quite important for one group of plants that I look at. That is the jacket, the pulpit family, the philodendron family, which actually play with this. Also in the magnolias, there is something going on in that direction as well.
Interesting, yeah. Not Terminal Flower, not quite as funny as Sonic Hedgehog, but it's all right.
No, no.
No Sega characters over on that end of the phylogenetic tree, I guess.
No, no.
But but the I guess the first question I wanted to field with you was, as you know, the origin of life. Figured we could talk about the origin of life, the origin of eukaryotes, and the mega groupings within eukaryotes. And so,
Hold
you
on.
from taking your
I do have
class.
a bit problems with the reception.
Sorry.
I do have a bit problems with the internet apparently. So
Um.
sometimes your text stops, so you have to say this again. What is the first question again?
Oh, the first question is, uh, well, I know that you have a bit of knowledge in the origin of life, and that's a very interesting topic, in my opinion. So we can probably broach that one first. You wrote here three essentials: energy flow, functional information, and compartmentation. So, would you like to explain what you mean by those with regard to the origin of life?
Yes. So one of the big debates about life is that life is always a state of high order. So you have molecules in a very sorted way. If you look, for example, at a protein, that is a very specific line of amino acids. Once you have that series of amino acids, they do actually fold in a pattern that follows the entropy laws. But how do we get this specific pattern and this specific sorting? And a lot of people debate that this could not happen by itself. That sorting would not happen because everything in the universe goes into a entropy stage.
Right.
And that is Earth not the case. Earth is not a closed system or an open system. We have energy flowing in and flowing out of the biosphere, out of the life system of Earth. The most famous energy flow that many people refer to is the sun. It's not the only one. The sun is a good example. Everybody understands that. So the sun sends energy into outer space, and 99.99999% of that energy goes into outer space and never hits Earth. Just that little blob that stands in the sun way, Earth gets hit by that massive amount of energy and absorbs that energy. Because energy comes to us as light, and most of that is in a pristine Earth, in a primordial Earth, absorbed by the surface and turns into heat. Another big part gets reflected into outer space. And that heat accumulates as a warm surface. You all know this from the warm street or the warm feeling on the sun. And that means that this light energy that comes in with a shorter frequency gets then expanded to a longer frequency, and that warmth is infrared. And in the shape of infrared, that light then dissipates back into the outer space. So energy comes in as visible light and leaves again as infrared. Life now steps in between and takes that light and lets it cascade through a sequence of metabolisms, starting with the primary producers like plants that make sugar out of this. And then these sugars, they basically percolate through life, through the first consumer, second consumer, till then the top predator dies and poops and all that stuff then becomes then garbage, debris, and gets broken down by the destruents. And at the end, it's all mineralized again as this energy cascaded through. And at every step, about 90% of that energy gets lost as heat.
Mm-hmm.
And that heat will then again dissipate into outer life, into the outer space as infrared. So that cascade of life, that is the flow of energy. And in thermodynamics, that is the work. With work, you can create more information. That is allowing you to reduce the entropy, to increase the amount of information in the system, at least temporarily, and on Earth, to increase then the order of molecules and create something like life. Once that energy stops, that energy flow stops, life will die. That is the flow of energy.
So
Next,
oh, sorry. Go ahead.
the next part is growth. Okay, growth is nothing else but actually accumulating molecules to that higher order. So if you have a concentration of sugar around you which is lower than what you have inside of your cell, by nature, the sugar concentration will go from the cell to the outside. The cell now, using energy, can accumulate that sugar into the cell and generate our sugar content inside of the cell than outside. That's eating. By eating, you can generate more volume in the cell, more biomass, and grow. So growth is nothing else but the application of that sorting mechanism using light. Eventually, we go to information. I mentioned already, energy allows you to build an information system that has a higher content of information, such as life in a group, such as membranes, proteins. Then eventually that information is stored in the form of inheritable molecules on Earth that happens to be DNA and RNA. And that can be duplicated and passed on to the next generation. That is then reproduction, which is also a feature of life. Reproduction is nothing else but multiplying that information using that energy-dependent growth. By doing so, basically using that energy flow to build more order temporarily, which we store as DNA and RNA. That is the principle of life.
Do you think that there was a molecule that predates RNA, for instance, something like 3-ose nucleic acid? Are you of the opinion that maybe one of these acted as a template and passed the information to RNA later?
Well, the part is RNA itself is a molecule that's a polymer of nucleotides. So before we can make RNA, you need to have single nucleotides. Nucleotides are relatively simple molecules. They have an adenine group and they have a ribose molecule, not the adenine group, but a nuclear base on it, like adenine, guanine. And then you have a ribose molecule, or in case of DNA, deoxyribose, and that's simply a sugar, and a phosphate. Now, phosphate is very simple to form, actually, just phosphoric acid in water. That happens from volcanoes. Then the Miller experiment has shown that simple nucleotides like adenine, guanine, nucleic bases, and sugars, simple sugars, can form spontaneously if you have energy flow and chemical reactions. And so what then happens is you have a polymerization of these monomer molecules. So yes, there were definitely the single molecules around before RNA formed. So RNA, if you have phosphorylated nucleic acids, it will polymerize based on the phosphate group added to them, more or less fast. If you have a catalyzer, that phosphorylated polymerization happens a bit faster. The catalyzer does not have to be an enzyme. A catalyzer can be a clay surface,
right.
It can be a membrane. It does not have a complex enzyme. Phosphorylation, this dephosphorylation happens on a lot of surfaces as long as there's a charge or a electric conduction on it. Platinum is a good example. Platinum is a natural, is very often used as a catalyzer. Catalyzes a lot of reactions, but it's by way not the only one. On small scale, even clay can be a catalyzer, clay platelets.
Right, that's why they talked about the clay theory, or the clay hypothesis of abiogenesis, the Morillionite. Yeah.
And even membranes, if you have a phospholipid bilayer membrane and a liposome, and we know liposomes can form spontaneously. We use this in factories to make cosmetics. A lot of cosmetics and medicine is actually administered using liposomes. These are basically, you can say, something very similar to soap in a way. They can form spontaneously. And if you have a high concentration of phospholipids, they will form micelles and liposomes. And if you generate this in a salt solution, and for example, because of rain, the salt solution of the surrounding of that liposome changes because it's less concentrated, you will get electric across that membrane. And in that only can be catalytic effective. And that's been shown with phosphoglucose. glucose. So phosphoglucose forming starch that has been shown that the membranes operate as catalyzers.
Right. Yeah, and it's fascinating how much research is coming out of labs on abiogenesis. Just seems like all the time it's a lot less difficult for these organic molecules to form than it was previously thought. Of course, also the
Yes.
geochemistry of the reactions has kind of changed as more has been realized about the ancient atmosphere and the ancient oceans and all that. But even still, it's still not that hard, it seems. Which
It takes less extreme conditions than presumed. At the beginning, in the Miller experiment, they had very extreme conditions with lightning imitating lightning and having open plasma. Today, we know we don't need open plasma, actually just certain surfaces to it. We don't have extreme temperatures. It doesn't even have to be boiling temperature in water. You don't need those extreme temperatures. And we do find those situations on Earth, actually. So it's not like
right.
it would not occur.
Yeah, it does seem though that the metabolism first hypothesis has kind of taken the lead, because as you're saying, you have to have a sort of an exchange of heat and exchange of molecules. Otherwise, you can't really get RNA and all the other stuff working. You got to have some constant flow of energy to get this all
Yes,
moving.
you do need to have a flow of energy, yes.
Yeah.
Very often, even today, living organisms, especially the chemoautotroph organisms that live in the deep sea vents or in the cold seeps, which are the sun-independent ecosystems, they simply sit into an energy flow that would happen naturally. They just use that energy flow to their own benefit. It's a little bit like if you have a river running down from a mountain, that water will run anyway, but you can build yourself a little power plant in between where the run of water drives your own little water mill. And then you employ that run of energy to your own benefit, but the water would run anyway. That's
Right.
what these organisms do. This basically the flow of energy, the stream of energy, sit in between and let their little own life mill run on it and profit on that. That's
Right.
what these organisms do.
Exactly. Well, speaking of the coal seeps, as if I remember correctly, it's the Archaeans who are one of the big, or big part of the flora of the coal seeps. And Archaeans are also play a big role in our own evolutionary history.
Yes.
Would you like to uh, illuminate how we are Archaeans? After all,
We are Archaeans. Yes, we are basically Archaeans that have rixetiosis. So our mitochondria are rixetias, but the DNA system that we have is basically an Archaean system. This is now a big jump where we talk from the original life. So we have been talking about chemical evolution and how we get from, say, free-floating nucleotides and free-floating phospholipid molecules to membranes and first simple RNA molecules, which are very, very far away from Archaeans. And then we jump a huge leap, go to a DNA-driven system with a complex enzyme system and a difficult, complicated membrane already, and then we call those Archaeans. And these are organisms, for people who are watching this, maybe not completely familiar for that. So there's two major principles of life on this planet. One are the bacteria, which many people have heard about, of course, and on the other side are the Archaeans. Like the bacteria, Archaeans don't have a cell nucleus, but they have also ring chromosomes. They look very similar to bacteria. They have simple chromosomes in it, and they look for the untrained dialog bacteria, and people in the old times described them as archaebacteria until they figured out that the Archaeans use a completely different system of DNA management. They have basically the same DNA, the same principle of enzymes reading the DNA into messenger RNA and using this for protein synthesis using ribosomes, but the ribosomes have a different weight. The RNA enzymes that make the RNA, there are more different. Bacteria have only one, Archaeans have many. The DNA has protective proteins that we eukaryotes have, but bacteria don't have them, and so on. And so if you look in greater detail, you see that the Archaean way of doing gene management is closely related to the way how we eukaryotes do gene management and what bacteria do. And so we are basically Archaeans, and that's what Archaeans are. Yeah. Pretty complex organisms if you compare them to simple proteobionts, so they are fully living organisms after the split from bacteria and archaeans. And at this time, it is rather, it's an interesting question if archaeans and bacteria have a common ancestor, and there is indication that yes, there was a common ancestor before archaeans and bacteria split from each other. And we haven't found that one yet.
I
It's not
read.
to my knowledge.
Well, yeah, there probably be no fossils of it, and even if there were, how could we differentiate them? But
Yeah, they're
the
all just little blobs, yeah, little coal blobs, yeah.
right, I read a paper a couple weeks ago that was saying the last universal common ancestor probably had an RNA genome, and that both bacteria and archaeans independently developed their replication machinery.
Into DNA, so both went into reverse replication,
Yeah,
reverse transcription, and came up with DNA as a long-lasting storage unit independently.
yeah,
So
that's
that is news to me, but that is a debate that I've seen the debate about it. But now that somebody states that, I don't know what the evidence for that is, but it's a good suggestion. It's certainly one of the suggestions that DNA evolved independently twice, and therefore DNA managements differ between two. Other people said there was a early DNA management, and then that diverged into the bacteria specification and the archaean specification. But if they have more evidence, that's good to know that something is.
that's Forterre, I believe his name is Forterre, who is doing that research. Sounds French.
Okay, that might be. And most of that probably is reconstruction using phylogenetics, generate a ancestral character state. And from my experience, it always takes time until that is really nailed in, but it's interesting, something to follow up on it. But that's one of the discussions. Did DNA evolve independently twice? Because it is a better storage unit. It is a better molecule to store information long-term. And it is not a very big step from ribose to deoxyribose. It simply take a OH group away from ribose, and you have deoxyribose. That can indeed happen twice independently. That is a convergent evolution that is absolutely possible. Yes.
Yeah, that'd be some. That would be some very, very deep convergent evolution, though. That'd be right at the, right at the start, like nearly.
Well, what I think is here the key part of the ribosomes. I don't know what Forterre says about ribosomes, but both units, both archaeans and bacteria employ ribosomes, which then do the job of translation into proteins.
The transcription and translation machinery, he says, is probably homologous, if I remember correctly.
Yeah, okay, exactly. And so the point is, the more you need to put all that information for ribosomes also in the chromosomes. It sits on the chromosomes as well. So that transfer is a bit more complicated. Coding genes, protein coding genes, translating those into reverse translating those into DNA is one story. Putting the ribosomal information into the chromosome as well, that would be a second story. And I think there might be some information in that. And I'm sure there's going to be more research on that.
Oh, absolutely. Yeah, it's kind of interesting because the entire clade, including eukaryotes, has to be archaea, or otherwise archaea is paraphyletic, because as they keep finding more and more and more and more groups, it seems like they're kind of all stretched out.
We can expect that the archaeans are paraphyletic. I expect this, at least. I would not be surprised if they would find that the extant archaeans that we have are not monophyletic. Monophyletic, for people watching this, means they have one common ancestor that is separate from the eukaryote lineage. My suspect is, as we keep finding archaeans and studying them better, we will find that it was one particular archaean who gave rise to the eukaryotes. This is just my speculation here, I admit this completely, but it is very possible that one of the archaeans gave rise to eukaryotes, and we just haven't found enough archaeans. The people watching this also say archaeans, they have been discovered relatively recent. There was already a student when they were discovered, that entire second domain of life. And they are understudied because many of them live in difficult to study areas. For example, in extremely hot or extremely deep areas, including very deep in the earth crust. So some of these archaeans live deep in the earth crust in very narrow cracks in the rock under extreme pressure. It's extreme high pressure down there. A human down there would melt like sugar, and they'll live down there, so-called extremophiles. And the moment you dig them up, and the moment you go down with a driller, you pull those archaeans up to our atmosphere, to our pressure, to our temperature, they die. Yeah, they bust and freeze at the same time, so to speak. They're not made to live at our cold, low pressure environment.
Right. There was
Study them well.
the Loki Archaeota, and it turns out it's an entire phylum that the Loki Archaeota are a part of. They also have, of course, Thor Archaeota, Odin Archaeota, and Heimdall Archaeota now.
So they used the Italian Norse mythology to name those archaeans. Why not?
They did, yeah, and they apparently are the closest relatives of the eukaryotes that have been thus far found.
Okay, that is interesting. So if they are, so are they still monophyletic with the other archaeans, or are they a side branch of the eukaryote branch now? That would make the archaeans paraphyletic.
Well, from what I've seen, the most recent papers I've seen, you have Uri Archaeota, which is the major Archaean branch. Then you have Depan, which is another group, and Tech, and then you have eukaryotes, and sister to that is the Loki Archaeota group.
So the Loki archaeans are sister to eukaryotes. So that is different phallogram that I saw last time. So now, indeed, that would change the story. Quite interesting.
I'll have to send you. I'll have to email you some of the papers I've read on that after this, because I have them all in like a.
They're stored in some way, yes. You have them all collected.
Yeah, I'll have to send those to you.
That is indeed, and of course, Loki archaea are now. I bet they're named Loki because they sit in hot, deep areas, right?
In Loki's castle, which is a volcanic region, underwater, near Iceland, I believe.
In Iceland?
It's kind of near Iceland, but yeah, it's this volcanic.
Is it under ocean? Is it under the ocean, or is it above the ocean? It's on Iceland itself. It's like outside.
No, it's real deep in the ocean, near Iceland.
Real deep, so outside, somewhere on the mid-Atlantic ridge, somewhere a bit south of Iceland. Yeah, that makes sense to call them Loki then.
Yeah, it's they got the whole pantheon there of Archaeans, but we are Archaeans because our ancestor did something very, very interesting, which is part of the sort of the contingent nature of history, which, it just so happened that a bacterium and an Archaean came together, and had this never happened, there would be no eukaryotes. We wouldn't be here today.
Well, there's two steps involved. One is formation of the nucleus. Right. And complete dramatic change of how we handle our DNA in terms of chromosomes. So different from Archaeans, eukaryotes have much more genes. We have increased our gene portfolio enormously. Get that many genes, that little ring chromosome that Archaeans and bacteria have isn't good enough anymore, doesn't work anymore. You get yourself a K-palette of chromosomes, and so at some step, those Archaean prokaryotes opened up the chromosomes into linear chromosomes and also evolved a new way to handle them, that is the spindle apparatus, centrosomes with the centrosomes in it, and that allows us to pull these chromosomes around, including sexual cycles, meiosis, mitosis, and cell cycles and stuff. And that is one very important step, and I think this is actually much more important than mitochondria. That alone changes the way how eukaryotes can manage their large genomes and recombine those genomes directly in a concerted community. According to Dawkins' selfish gene concept, if you have only few genes, you can change one or two without messing things up. But if these genes are very strongly interdependent and still organized, you cannot just move one gene easily and expect that things are still working. It's like in a car, if you have a little lawnmower or a go-kart, you might play around by taking one spark plug out and taking another spark plug from another place in, and it works. But if you have a Honda Civic, then spark plugs are more sophisticated, and just taking a fourth spark plug into a Honda Civic spark plug might not be the right choice. And so part, you have then to move the bits and pieces in a group, means you have to change larger chunks of your genome, and that is where sexual reproduction comes in. Eukaryotes can do that. The other part is the mitochondrial invasion, and that is something which gave probably the eukaryotes a very big boost in energy production. But whether this was necessary, we do not know. So
Right.
that's a second step on top of the formation of the entire nucleus and spindle apparatus. And it's pretty well resolved what happened there. So we know that Rixetias are the sister bacteria of our mitochondria. We have discovered that. Makes a lot of sense from DNA analysis. It also makes a lot of sense.
Whoops. I think whoops.
Can you splice them together?
I've never tried that. Let's see. It's so funny because we. Oh, okay. Can you hear me now? Okay. Well, it says reconnection successful.
I hear you now. Yes.
Okay. Well,
it says your bandwidth.
Oh, I'm sure it's my. I'm looking online to see if it's how it's affecting the live stream. Oh man.
We may have to repeat this if it sucks so bad.
Okay. Well, yeah, we can.
Accuracy of the creationists. They step on a line.
An alternative solution. I streamed through OBS, which is probably part of the problem.